in 2021. In 2021, there are 20 papers so far. And maybe he didn't even know that. I'm so grateful he's taken a little bit of time to join us here. He's the Rachel Carson Professor of Biology at Yale. He was formerly chair there for 7 years. He's also chair of the Diversity Committee, among all kinds of other wonderful diverse service. He began his career at University of Rochester and then moved to Michigan State. Like so many of the leading biologists of our time in infectious disease, he and Bob Woods both studied under Rick Lenski, who's inspired me as well, although my field is very distant. The inspiration is still there. He took 3 postdocs, one at University of Maryland, one at University of Valencia, Spain, and one at National Institutes of Health, until moving to Yale in 2001. And he's been there ever since. And they've very much appreciated him, as have all of us. Does everybody here know that the World Health Organization says that the greatest threat to human health in 10 years will be antibiotic-resistant infections? And it's also very, very clear that we are not going to invent new antibiotics that outrace natural selection. Paul's work, along with that of some other people, has been looking at the natural ways and the competition between viruses and bacteria, phages they're called. And I have always seen this as an extraordinary opportunity. Unfortunately, and I'll ask Paul this later, I mean, you can't have a drug company give you $1 billion to do this because the products are not readily patentable. But nonetheless, extraordinary things, especially in Russia, where he went for a visit in 2001. I'd really like to hear about that as well. He has all kinds of honors and awards. I'll simplify by saying that in 2019, that 1 year, he was elected to fellowship in the National Academy of Sciences, the American Academy of Arts and Sciences, and fellowship in the American Academy of Microbiology. Ben Chan joined the group there a few years ago. He took his degrees at University of Utah. He then worked in industry for a year and then joined the Turner Group in Yale at 2013. His focus also has been developing phage therapy. This is such exciting work. I won't say another word, and I'll turn it over to you, Paul.
Wow, thank you so much. It's a pleasure to be here today. I appreciate that introduction, Randy, and thank you all for being here to participate. Indeed, I want to show some slides, but the idea is not to dominate the entire time because it would be wonderful to have conversation with you all. So let me go ahead and share this. Okay, hopefully that's full screen and you can see that. So let's take the next 20 minutes or so, and I'll go through background that is also covering some of the recommended reading for today. And I know that not everybody had a chance to do the reading, so I'll bring you up to speed there. These first 3 pictures, I often begin my talks recently when I present on this project. From the left to the right, this is very indicative of what we're trying to do. It is very possible to tap into the Earth's natural biodiversity, which is indeed vast, for bacteriophages of different types and particular biologies, to think about how this could help remedy the problem that antibiotics no longer address. So when you see Ben Chan in my group there on the left, sampling from the wild, this can quickly lead to phages that are characterized in our laboratory, as shown in the middle picture. And what I'll talk about today is, you'd be surprised, I certainly am surprised, how quickly it could lead to personalized treatment of, in this case, a young woman who's in a hospital room in Texas, and she's being shown a vial of phages that have a natural source, and that these would be used ideally for her to inhale them over a multi-day period to try to have those phages destroy the bacteria that have invaded her lungs that are multi-drug resistant. In fact, in her case, she's dealing with a pan-drug resistant infection, and she happens to have cystic fibrosis. So we'll hear more about her later on. To begin, I have to disclose that I'm a co-founder of Felix Biotechnology, a company that seeks to commercially develop phages for therapy. Here's the topic today, the antibiotic resistance crisis. As Randy alluded to, this is the quite abundant evidence that an increasing proportion of bacteria are resistant to the commonly used antibiotics in biomedicine. And this is leading to the problem of this projection map in the upper right, is that a very short time period from now, a few decades from now, it's expected that this would be a leading cause of death in the human population. And indeed, it's a worldwide, it's a global problem. Big Pharma is not as interested in developing antibiotics simply because it does not make money, and I can understand that. So if these are developed and found and brought to market, this takes an astronomical amount of money, And this is an investment that could lead to something that's literally placed on a shelf as a last resort drug, and therefore the return on investment is not very much. So many people have started to call this a discovery void, that we are no longer turning to big pharma for antibiotics being discovered. So there are alternatives. This is a very old one. It's called phage therapy. It has been developed in many countries for over 100 years, including in Russia, Georgia, the country of Georgia, Poland, etc. And more recently, Western science is looking at this idea because we are running out of other options. The cartoon on the lower left is a reminder of a lytic phage reproduction cycle. These are often the phages, these types of bacteria-specific viruses that are sought for these alternatives to antibiotic drugs. And the reason is that you can see from this life cycle, this reproductive cycle, that the phage instructs the bacterial cell to make copies of these viruses. So this is essentially a self-replicating drug, which is highly attractive in terms of minimal dosing and other benefits. So you have specificity to the target and you have the self-amplification. That said, there are some obvious costs, longstanding costs of phage therapy that are on our minds. If you have too much specificity, this gives you too limited spectrum. And it is indeed the opposite of a broad spectrum antibiotic, which is itself problematic in many ways, but this is the opposite of that. The delivery to the infection site could be easy or difficult. We can talk about that later on. The immune recognition of the human immune system to catch on to the fact that there may be very many phages in your body that your immune system is ignoring, but if one is administered to you at very high dose for a multi-day period, will your immune system catch on, produce antibodies against it, and therefore you can't have this move forward. But last, as an evolutionary biologist, I care about the fact that we have abundant evidence that phage resistance is easily achieved in bacterial populations. So you have to think about this being a therapy that is in many ways destined to fail. And this is why we try to think of it as an evolution problem and how that could be overcome. Here's a quick reminder of the very many ways that bacteria can evolve resistance to phages through their various defense mechanisms. We are literally finding more of these every day in research. On the left are more of the classic ones that have been known for some time: barriers to entry, recognition on the inside of the cell, such as restriction enzymes that cut up recognized pieces of foreign material entering that's nucleic acid. And those can change evolutionarily over time. But on the right, these are the more recently discovered mechanisms. They're more elaborate. This is CRISPR-Cas, where a bacterial population can build up recognition cassettes against common common phage predators, if you will, of that population. And you get this buildup of recognition to fight them and to recognize them and defend against them. So we can talk about that more later on. But here's the real crux of the problem. If you consider that phage resistance occurs readily in bacteria through evolution, can we take advantage of something else that we see in evolution all the time? In other words, can we pick the right phage to exert the right selection pressure? So keep that in mind as I now describe to you what an evolutionary trade-off is. Here is where we see in evolution that very often evolutionary change involves compromises. So as natural selection works on any population, bacteria, humans, or whatever, to evolve traits to improve performance, this could lead to other traits that don't function quite as well and have some liabilities built in. So I want to give you a quick example from humans. What humans arguably do better than any other animal is hurl objects with accuracy and speed. And the best of us do this professionally as baseball players and softball players. But we are the only bipedal great ape. And so therefore we have musculature that allows us to free up our hands to do work and to hurl objects with great speed and accuracy. But walking around all the time on two legs is quite different than what chimps and gorillas and other great apes face. And for us, this creates a liability, a built-in liability, that our lower back pain is something that comes along with being a human, and also neck pain. So this puts a lot of strain on our bodies when we walk around all the time on two legs. Now think of this concept carrying over to phage therapy. Our goal was to not choose any old phage for phage therapy. Often historically it's been find a phage that will kill an important infecting bacterium, and classically that has been vitally important and has moved the field forward. But we want to think of it with a greater nuance with an eye to evolutionary biology. If you pick the right phage, recognizing that it can do the classic job of killing the bacteria, depending on what that phage is doing to interact with the cell, it's also going to exert selection pressure in a particular way for those bacteria to evolve resistance. And this could be advantageous for us in the therapy. Two quick examples. This phage here in red is interacting with this hypothetical bacterial cell by binding to an outermost protein of what's called an efflux pump. This is a protein complex that if the antibiotics get in to certain bacterial pathogens, those antibiotics are actively pumped out so that the bacteria are able to gain widespread resistance to antibiotics. If you have a phage that is interacting with those complexes of proteins, especially by binding to the outermost protein of the complex, then this could create a huge problem for the bacteria. They're going to die from phage attack, and they will be selected to resist those phages by altering or eliminating the efflux pumps, therefore shifting the remaining bacterial population that is resistant to the phages to being sensitive to antibiotics instead of resistant. Obviously, a better outcome for biomedicine and for phage therapy, in our opinion. Here on the right is another example. I'll go through it more quickly. This phage in purple is interacting with an outer capsule of this bacteria that is allowing the bacteria to be shielded from recognition by cells of the human immune system. So if you use a phage in this way, it will select for the bacteria to ditch the capsule or change it, and therefore you can get the classic killing of the bacteria as well as selection for lower virulence because the bacterial population left over as mutants change in that way. So how would you find such phages? I won't go you through the long pipeline of discovery that we go through, but here's a very key node or a key point in that pipeline. It's not that easy to figure out what any virus binds to on the cell surface. And the same goes for phages. So we've tried to develop a more high throughput way to find phages that are interacting with certain outer cellular properties that we care about that we think could work in that trade-off that I just described. So one of my former students, Katie Courtwright, who is now a postdoc, she used existing in-seq technology where you use transposon insertion of bacteria, you expose them to a phage environment, and what you'll see is outgrowth of the bacteria that have the genes knocked out for which that phage is utilizing as important in its infection, especially in the binding to cells. So we're looking for information that shows up in this hypothetical blue region of this graph shown below. And here are two examples. If you take phage T6 of E. coli, it's classically known to enter and interact with those cells through the TSX porin. So what you see here is this screaming high, statistically significant hit when phage T6 is used as the selective factor in this analysis. So that's confirmation of something that we already knew. Here on the right would be a very different example. This would be a phage that we've called TIVP-H6. I'll describe it more in a moment. It infects P. aeruginosa, Pseudomonas aeruginosa. And what we found in the same analysis is that there are lots of type 4 pilus genes that are important for the construction of that feature on the bacteria. And if you knock out any of those, you get good statistical hits for what this phage is using to bind to the cell. And that tells us that the type 4 pilus is the way that that phage is very likely interacting with the cell. And that's going to become important in this slide. So if you find such a phage, this is an example of how it can select against virulence. And that's because type 4 pili, indeed, these are important for lots of traits in these bacteria, but they are also contributing to the production of piocyanin, which is a pretty nasty virulence factor. This happens through an underlying gene network that I don't have time to describe, but just understand that this virulence factor causes toxic effects, inflammation, and oxidative stress. So if you had these bacteria incapable of forming the type 4 pili, then this would be a way to avoid the phage attack, and it would also cause them to be worse at producing this virulence factor. So here on this color picture on the left, when you grow these bacteria up as wild-type pathogens and you spin down the cells and look at the supernatant, this piocyanin is produced often in abundance, and it has this blue-green color. Here on the right would be an example of a phage-resistant mutant that avoids this phage attack, but it is debilitated for its ability to make this virulence factor, and therefore it looks just like regular growth medium. So the last thing I'll say about type 4 pili is that they're also important in producing biofilms and just the movement of bacteria. This is called twitching motility. So there's lots of traits that would be debilitated for a bacterial pathogen if you're selecting against type 4 pili like this particular phage does. So I'll come back to that example when we look at some clinical evidence. Here's a quick bit of some unpublished data on this particular phage. If you take these P. aeruginosa bacteria and expose them to various types of antibiotics, you'll often see that those antibiotics are inefficient or incapable of killing the bacteria. And what's being measured in this graph is the production of thiocyanin. So unfortunately, the exposure to some antibiotics, such as Cipro in this example, will cause the bacteria to not only avoid dying from that antibiotic, but they'll actually jack up their production of a virulence factor. Erythromycin could be something that will calm the virulence factor thiocyanin problem, but it wouldn't necessarily do anything to kill the bacteria. Here on the far right is a phage that does both. It's killing the bacteria. And what these red data are supposed to emphasize is that in the after the fact, that the leftover mutants of the bacteria are very debilitated in producing thiocyanin. And this ability of the phage to reduce that trait, this works very well in combination with erythromycin that I mentioned earlier, and even in the presence of Cipro, which was having the opposite effect. And now this is a much better outcome if you say administered it alongside this phage. All right, let's look at one other quick example now of a phage that we took straight from nature, and it selects against antibiotic resistance. This is a little more to the issue of the antibiotic resistance crisis. So these are data from an E. coli model, and this was a project that was led by one of my postdocs, Alita Burmeister. And again, this is an example of a phage that is interacting with an efflux pump. In this case, it's binding to the outermost protein of the Toll-C efflux pump of E. coli. Here is the consequence of if you grow these wild-type bacteria and a Toll-C knockout in a phage-resistant mutant, one of the resistant to this phage, U136B, in a non-antibiotic environment, they grow similarly. But in the presence of a particularly high level of tetracycline, you find that the Toll-C knockout is unable to grow as expected because its efflux pumps are not moving that antibiotic out of the cell efficiently. But you also find that this phage-resistant mutant is debilitated for growth. In other words, the phage is indeed killing the bacteria, and it leads to selection for resistant mutants to the phage that are debilitated for their ability to grow in the presence of tetracycline. Here to the right, we now know that this one phage binds to more than one thing. It's not only binding to Toll-C efflux pump protein, but it's also binding to the lipopolysaccharide, or the LPS, that is surrounding the cell. So this is a side view of a diagram of LPS, and here in the middle, I've colorized with these rectangles, the portions of LPS that we now know that if those are missing, the bacteria can't make that portion of LPS, and it also prevents the phage from binding. And here to the right is the consequence. So the wild-type bacteria and various mutants are compared for their growth, and what you see is the wild-type is able to grow in the presence of colistin, and this axis here is increasing colistin sensitivity. So now in the presence of a different kind of an antibiotic, these phage-resistant mutants have a problem. They have increased their sensitivity to this other antibiotic, colistin. So essentially one phage is driving two trade-offs. So one more bit of empirical data before we look at clinical evidence. These data are from the work of James Gurney, who is a postdoc who's working at Georgia Tech, and we've collaborated with him. He's used a wax moth larvae model for P. aeruginosa infection, where what he found in the study that we collaborated with him is that phage OMK01, this is one of the first phages that we talked about in its ability to drive trade-offs in P. aeruginosa. Indeed, when you administer this phage and you especially allow it to synergize with an antibiotic, you find that you have much better ability to keep these wax moth larvae alive in the face of lethal bacterial infection. And here to the right is again the evidence of the trade-off now in an in vivo model. In the presence of phage or phage plus antibiotic, indeed this seems to drive the evolution of resistance to the phage that leads to an increased sensitivity to particular antibiotic, in this case erythromycin. So now I want to finish up by going quickly to some clinical evidence before we turn it over to discussion and questions. OMK01, that last phage that I just talked about, we used it some time ago and presented the results in Evolution Medicine and Public Health Journal that we had the ability to do US FDA approved treatment of a chronic biofilm infection in a man who was suffering from an aortic arch replacement that became infested with a multi-drug resistant P. aeruginosa strain. So here to the left is a reminder of what an aortic arch replacement is. And I turn you to, or I suggest you go read the paper for details because I don't have time to go into it in great detail. But this was just a fantastic outcome for this man who was essentially slowly dying from this infection that by the time they had opened up him to find out how bad it was, it had eroded away part of his sternum. But by administering this phage plus a useless antibiotic, they synergized together to be able to cure this infection. And he was able to go back to work at an advanced age, but he was very willing and able to work as an ophthalmologist here in the New Haven area. So this was talked about a lot. This and other work has prompted articles like this one in Scientific American from a couple of years ago, where I want to emphasize that, okay, phage therapy might be here to stay in Western medicine, but it never went away in many parts of the world, as I said earlier, and I believe that Randy said at the very beginning, in places like Russia, Poland, and the country of Georgia. It's more about what are we doing in Western medicine now that might be a bit more in agreement with what has been a longstanding tradition to try and develop phage therapy in those countries. So where can we go with this? So I'll now show you some unpublished data referring back to that young woman who I showed you her picture earlier from the hospital bed in Texas. So she is a 22-year-old with cystic fibrosis, and anybody with CF has this greater vulnerability to infections of the lung due to the physical structure of their lungs associated with that genetic disease, the widening of the airways and the overproduction of mucus. So in her case, she had, unfortunately, a pan-drug resistant infection of P. aeruginosa when she came to our attention. Here to the left, a few summary statements of her problem. Really in the middle here, you see the data in green. This is FEV1 data. How much air can she force out of her lungs in the first second when told to do so? And this is a way to gauge lung function in CF patients. It's very widely used. But what you find is in a multi-year period, she had shown the sharp decline in lung function. And arguably, she's undergoing pulmonary failure, at least pulmonary decline, if not failure. And worse yet, her infection here to the left, these red R's are referring to the fact that all of these antibiotics were not working for her infecting pathogen. Now, we administered therapy here to the red arrow, the date, and immediately through adding phages to her nebulizer, and she breathes this in as a way to deliver the phages to her lungs and try and treat that infection. This worked amazingly well and over a multi-day period. And after the fact, you see the trajectory of her lung function improved. And this essentially caused her to avoid a lung transplant. Best yet, when we summarize her metrics after the therapy, we did not remove the infection entirely, which would be a very difficult thing to do in a patient like her who has CF, but the leftover infecting bacteria were largely now sensitive to antibiotics and demonstrated this trade-off that we saw in vitro. So this is excellent news for her and her physicians that they're able to deal presumably with this problem in the future if she has what's called an exacerbation or a worsened state from that infecting bacterial infection to outgrow. So I can't summarize all the very many people that the folks that I've worked with have been able to help. I think to date, we've done roughly 20 chronic infections, most of them pulmonary. They've been treated either here at Yale New Haven Hospital or at partner institutions around the USA. I'm summarizing here roughly 13 or so, I think, of the first cases. I'll highlight again that many of these were CF patients, but you can also think of non-CF bronchiectasis and chronic obstructive pulmonary disease, leading to vulnerabilities of the human lung that are quite similar to CF. And therefore, we see these individuals with similar problems and have tried to help them. So the data that we are amassing and excited to present soon in a case series paper, let's just consider for the CF patients we treated, there's roughly 9 of them. Again, they're getting nebulizer treatment, but we're able to take sputum samples that they provide and examine those before, during, and after the treatment. And we can come up with some excellent summaries that go in the directions that we hoped. So does their lung function improve after phage therapy? Yes. Does their bacterial load, as evidenced from the outgrowth of bacteria in their sputum, decline? Yes. Do you get this antibiotic resistance flipping to sensitivity when you administer a phage that hopefully is driving such a trade-off? Yes, in many cases, that's what we see. So I'll take a closer look here at the inflammatory molecule reduction that is often quite evident. Here, what would happen is if we administered a phage to a person who's having a P. aeruginosa infection, where we're trying to toggle this virulence factor piocyanin and have it go away so it's not so destructive to human tissue and exacerbating the problem. So you can take clones from patient sputum over time, as shown in this graph for a single patient on the left. And what we often find is multiple clones taken at different time points, but essentially piocyanin production as measured in the laboratory declines and troughs out so that it's looking at the same level as just growth medium, where there's no piocyanin evident through that measurement. Here on the right is the consequence. If you take those clones, grow them up in the laboratory, and then look at the supernatant, put that on airway epithelial cells of human origin, and you see how much stress or inflammation is this causing in those cells. And you'll often find with markers like IL-8 and IL-6 that there's a sharp reduction in this, and this is further evidence that the trade-off is going in the direction that we hope. So a lot of this is predicting what's happening in the black box of the human body. A part of my work that I'm not going to be able to talk about today, but is impacting all of our lives, is the emergence issue, and especially SARS-CoV-2. So we, like other laboratories, are trying to better predict the emergence events and pandemics of the future. And this is, of course, very difficult business to do. We've taken the angle of, is there enough evidence that we know already that when viruses infect multiple host species, it's easier for them to jump into humans and cause problems in just one more host, meaning the human species. So those are the sorts of predictions that we're making and examining in directly infectious as well as arthropod-borne pathogens. And we're trying to also look at the importance of SARS-CoV-2 in animal reservoirs that might see the virus. But here's the relevance back to what the topic is today as I finish up. We're trying to find phages that are very good at causing infections to go forward in genotype space of the target pathogen. So in other words, we want them to be as generalized as possible. But we are wary of the fact that if they are highly generalized, they might be also poised to emerge, if you will, on other species in the microbiome. So this is somewhat similar to our work on emerging and pandemic pathogens, is that we want to be able to predict what are the rules governing whether or not host species jumps are going to occur. So the next steps that we have are very many. We're looking at currently a clinical trial here at our hospital, abbreviated as Sci-Fi, and it's essentially looking at phage protection of the CF lung in healthy volunteers over a multi-month period compared to those receiving nebulized placebo. We have now the resources for a Center for Phage Biology and Therapy here at Yale, where we want to continue developing such projects, and we would like to work more and more collaboratively with those who are of interest to move this forward. Also, I have to thank my team and people like Toby Nagel, who has Phages for Global Health as something that she's passionate about, is taking this knowledge to others in countries where they might have more humble resources to obtain drugs now and in the future. So it could be teaching people now about the pros and cons of phage therapy and phage biotechnology is something that will give them a head start for something that could be a very affordable and more widely approved drug in the future. So I'd like to end by acknowledging the many folks who actually produced the data here, my current and past lab groups, and the people who actually contributed to the data that I presented today are shown on the upper right. I'm mentioning also Deepak Narayan, who was the surgeon who helped us with the first patient case. So I'm quite grateful to him. He's no longer alive. We lost him a few years ago, but without his belief in what we were doing, we wouldn't have been able to take this technology to help others. I'd like to thank the funders and also thank Rachel Carson for her amazing contributions to science. I'm very proud to be the Rachel Carson Professor at Yale University. So should I stop sharing? Maybe I stop sharing, we'll move on that way. Hope that's okay.
Yeah, I think that's great, Paul. Thank you so much for a great presentation.
Thank you. I almost ended on time.
We're fine. This is we're in a good place here.
Great.
We're going to turn things over now to Bob Woods as our clinical discussant, which is something we try to have at many of these Club EvMed. So we'll turn things over to Bob Woods, Dr. Woods, for a couple of minutes here.
Yeah. Thank you, for inviting me to talk about the clinical aspects, although Paul's already presented actual clinical data, which is sort of really, I think, a testament to how impressive this body of work that he's kind of led and been a part of taking this from basic science all the way already to treating patients. But I will, I think, in terms of putting this into context from the perspective of an infectious disease doctor, I think thinking about where do we need phage therapy, where might you use phage therapy? You mentioned cystic fibrosis, but in general, we need new therapeutics where our current therapeutics aren't working or not working well, right? And for infectious diseases, right, where antibiotics fail, it tends to be in these multi-drug resistant infections, but also in infections that are anatomically complex, like such as the CF lung, but also infections involving the heart or bone, large wounds, microbial infections, and importantly, infections that involve mature biofilms, which antibiotics are do a very bad job at eradicating currently. And so thinking about where we need the phage, and in fact, where they've been used, mostly in the published literature, kind of let you know how what we're up against. For one, it's a high bar phage therapy where we're using it in the hardest possible cases in general. But importantly, it means that we're using these in settings where we would expect to see this evolutionary dynamic that Paul just talked about, right? It's that we're using it in cases where the direct killing effect of the phage is probably not going to take care of the infection, right? These large, complex, often genetically diverse infections. And so I think that's why thinking about these dynamics, studying these evolutionary dynamics is really key. And so prescient, think about the clinical impact. And the work, such as the papers that you presented, are really sort of foundational for this. But I think moving forward, and the sort of couple slides you included at the end of the unpublished data, I think that's where we're sort of excited to see what's coming next, because if you're proposing to use sort of these evolutionary trade-offs, sort of our understanding these evolutionary dynamics, those are things that might not translate well from our lab, from a model infections to human infections, where the population sizes are different, the diversity is different, the ecology might be quite different. And so I think I won't talk much more, but maybe if it's okay, start with a question, which is, given that you sort of have these concepts of these trade-offs and how to leverage these and some really impressive preliminary data, how do you design these clinical studies of the patient use of phage, so you can actually demonstrate that these evolutionary dynamics are being used, could be used to better therapeutic effect?
Yeah, great question. I mean, this is the tricky thing, is not only is the personalized treatment, the emergency cases, difficult to design because there's not much of a roadmap for it. But I took your question to also mean the very different goal of running a clinical trial in healthy volunteers. What is the match to the emergency cases, to the kind of a person who would be able to participate in that trial, and what does one measure, right? So the thing that we often see in phage therapy is that it does work. Historically it's shown to work, but mechanistically there were very poor tools to understand why, and that's not really an excuse in the current day because we have much better tools. But the last thing I'll say, because I don't want to dominate, is that what I'm really excited about is in these treated patients, as well as those who are volunteers in a trial, the power of longitudinal analysis, especially to measure things that should be indicative or not of these trade-offs happening. And to be clear, we see the evolution thinking and the leveraging of these trade-offs as a huge bonus to have the therapy work at all. But it's an enduring mystery that suppose the therapy works without that bonus, right? So that is more of a indicator that maybe this therapy works better without, not without, but on its own, without pushing too much to have particular phage characteristics. I kind of doubt it. I think the more that we know characteristically of the phages and understanding mechanistically how they're interacting with the bacteria is going to help us. But I'm admitting that it's an awesome time to generate the basic research outcomes, because so much of this is really unknown.
Okay, that was great. We also want to give a chance for some of your collaborators who are on the call, Paul, to speak up if they have anything that they'd like to add, or if you have any thoughts about the question that was just posed by Bob Woods. We have Ben Chan is on the call, James Gurney. I think there may be some others as well. If anybody would like to speak to this question that was just asked, or provide any other insights to what was presented.
If Ben's here, he's definitely the expert more than I am. So if he's there, he can definitely take that question.
Well, why don't we hear from you, James, if you're willing, while Ben figures out his camera. I think that's what I'm missing.
I mean, yeah, I haven't designed clinical trials, but
maybe let me just, I'll ask it of you.
Yeah,
I mean,
sure.
you've designed in vivo experiments with the wax moth larvae.
Mm-hmm.
You know how, I mean, it's even unclear to me in animal models. You know, how do we take a lot of the successes through time?
Yeah.
And let me just throw it out there, is some stuff could be revisited. There were classic experiments for which we didn't have as good of tools, and they need to be done again, right? And I'm not saying your work is redundant that way, but I like how you've used a new model that's a more affordable one for many researchers to explore the problem. And maybe you want to say something about how easy or difficult it is even to design those experiments.
I mean, that experiment was entirely predicated on two things: was the power needed to sort of really identify these survival effects, and the ability to have a quick turnaround for Pseudomonas. I'm a CF researcher for other people listening. I'm a CF postdoctoral fellow, so a lot of my work has been focused on translating these ideas into cystic fibrosis, and there are very few decent animal models. Almost all the infections are acute based, whereas CF is obviously a chronic disease. So I think, yeah, I like to say when I give talks, I like to say, you know, I don't want to be here when I'm 50, 60 years old saying, well, we tried in 2020 and it didn't work this time. They tried in 1990, it didn't work this time. Phage therapy only has so many more false starts. So designing these experiments and making sure we have the appropriate controls and the correct background, and to know why failure will fail, therapy might fail. Like that's super important. Said, like the phage burn trial in the EU was a massive failure for understandable and predictable reasons, and we can't keep doing that.
Where one of those reasons was the actual phages being used in the storage conditions led to much lower titers or densities.
It was the titers, I think, were 1,000-fold lower than they thought they should, than they were hoping to add, which is basically just adding saline at that point in some cases. So
yeah, very difficult problem. I see that Ben has joined us, and I'm not sure, Ben, in his, he was just at another podcast, and he's podcast jumping, so he jumped over to us, and thank you for being here. And I'm not going to steal Bob's thunder because I think he's the discussant, but Bob, you may have to pose to Ben a little bit of the question that came up before, because I'm not so sure he was here.
Sure. I'll just rephrase what I said before, which is that in these complex infections like a CF lung, right? You guys have done a lot of nice work to show that these evolutionary trade-offs can be leveraged, and in the lab they are leveraged, right? That phage resistance may make you more sensitive to the antibiotic, for example. How do you think about, now that you're moving to all these patient studies, how do you think about designing those studies, and also how you sample those studies and interrogate and get the data to show that these evolutionary trade-offs are in fact leading to better therapeutic effect than just the direct killing effect of the phage alone?
Oh, tough ones. So the trade-off specifically, I guess. How are we designing stuff to do that? Well, I hope we're doing that successfully. I don't know though. It's tough, right? Because so for clinical trials, you have to have like, the primary endpoints, secondary endpoints, whatever. And for the one that we're running, the primary endpoint is just bacterial killing, because that's like a very simple one to measure, right? but a secondary out, so it's in CF and Pseudomonas killing. I don't know if Paul mentioned that, but we're running a clinical trial in cystic fibrosis to treat those with Pseudomonas infection with phage. And so the primary endpoint is bacteria killing. So that one we're pretty confident we can meet. But the hard part is I think some of the trade-off stuff, right? The clinical benefits, therapeutic benefits of some of these trade-offs might not be so trivial, right? Like we can reduce the inflammation inducing capability of the bacteria, but I think in CF, it's not such a simple, like one organism causing inflammation fixed sort of problem. But we have, I mean, the study hopefully is designed well, and that we're collecting a bunch of sputum, and so we can sequence, we can see the genotype change. We're doing a bunch of microbiology to see, make sure the phenotype is following what we predict in the lab. And then with the hope that in a lot of these complex cases, you can pull something out of the clinical noise that looks like a therapeutic benefit. Yeah, so I guess to not answer your question, there was that.
Okay, thanks. We have a hand raised by somebody who's identifying as Joe Graves, although I think it might not be Joe Graves because I see another Joe Graves. But whoever it is, we'd love to hear from you.
I apologize. I've clicked the wrong link here. I'm Kelly Spurgeon from North Carolina A&T. So I just had a question. I'm not familiar with, I'm very new to biofilms, and I'm wondering if there's a potential for phage therapy to, I guess, capitalize on bacterial competition, like weakening systems through other routes so that, competition is the main killer of those primary infectious bacteria like Pseudomonas. Is that something that phage therapy could be potentially used for in the future?
So if I understand the question, are you asking whether you can tip the competition, the battle in favor of something that you would rather win out in competition than, say, the pathogen?
That's right.
Okay. Yes, I think that that is plausible, and many people are thinking about that possibility. I would include that in the very many compelling ways that people consider how can you interact with microbiomes and prune things out with phages. And there's lots of excitement to do that for very many things. But I don't know, Ben, do you have anything you want to add about that? I mean, biofilm, this is tricky stuff when you're considering, it's kind of like a second order effect. If you want to have the phage perform some thing in the target community that leads to another effect following it downstream, I don't think we've explored that too much. I don't know if I want to speak up for Ben. He has much more of the finger on the pulse of everything going on in the lab. What do you think, Ben?
Well, that's a really cool idea. I think we haven't, from what I know of what we've done, we just have worked on disrupting biofilms with phage or preventing them from forming, and then I guess hoping that disrupting that community might have some positive benefit. And then, but I don't think, I know I haven't, but I don't know, someone else in the lab might have looked directly at measuring the competition and how that might be capitalized on. But so far, I think we've just been focusing on, disrupting it, and yeah.
Mm-hmm. Good.
Thank you.
Okay, thanks, Kelly. I'm glad you could join. Terry Taylor, you have a question. Please go ahead.
Thanks, Charles, and thank you to Professor Turner for a very clear presentation on a subject which my knowledge is tiny. But some of the remarks you made, mine is more about a research policy question rather than the detail of science. And that is, you mentioned the history of the work in this area, over 100 years worth, but being sustained more in Eastern Europe, for example, Russia, Poland, and you mentioned the country of Georgia. I wondered what is driving this, the maintenance of it, and something that occurs to me is multi-drug resistant tuberculosis, which is in those countries you mentioned, and course, in Asia, parts of Asia, which is rampant. What is driving that research there that's keeping it going and maintained? Are the lessons for us here in the United States from this perspective?
That's a great question. I would love for others to chime in here. My brief response is that historically, there are very many places in the world, including in the US, where phage therapy was explored in the early days of phage discovery, but it became relatively more popular in the USSR and Poland and places where they were not immediately investing in antibiotics after Fleming's accidental discovery of antibiotics. And then it started to enter into this difficult historical time period where there wasn't much respect in the USSR and the USA for what each other country was doing. So during the Cold War, I think there was almost a reason to reject anything coming out of certain parts of the world just on principle alone. So that led to a slowdown. Now, in the current day, I think the last thing I'll say is that budgets can be quite slim in some areas, and they have to continue to invest in things that they had invested in the past that show promise. And it's more about, can they keep the funding going in places like the George Eliava Institute in the country of Georgia to get as much done as they had historically done in the past. Anybody else have anything to add to that? Okay. Just want to know if there's any phage therapy historians involved.
Thanks for that question, Terry, and really interesting. We'll just jump into the chat really quickly before we go to the next question with a hand raised. Jesse Meyer had a question. First said, great presentation. And then I was wondering how we can be confident. Jesse, are you on and do you want to ask your question? Okay. Would you like to ask it? Great.
Yeah, sure. So I was just wondering how we can be confident that phage therapy would decrease antibiotic resistance rather than increase it. I saw in the suggested Burmeister paper that they tested multiple samples of E. coli, all started in identical conditions with phage, and they saw pretty widely varying results of phage and antibiotic resistance mutations, even some samples actually increasing the resistance to antibiotics. So I was just wondering, when you give somebody phage therapy, how can you be confident that it won't, do something bad?
Sure. I'm going to answer it first from the in vitro side, and I'm going to leave it up to Bob or Ben or somebody else from the in vivo patient side, which is the harder answer. So on the in vitro side, it's just a numbers game, right? So I can't control, no researcher can control what the bacteria will do spontaneously through mutation. But I think what we can explore through population genetics models that are also incorporating fitness effects is how much do you really need to worry with the spectrum of possible genetic results about what is most common versus least common. And that gives you the confidence to go forward that at least on average, things are moving in the direction you want or the state space you want with the phage resistance mutations. So that's kind of what was explored in that Burmeister et al. Recommended reading. Now I'll turn to anybody else who's brave enough to answer it from the standpoint of decision-making for patient treatment. Yeah, what do you think, colleagues on the call? I feel like I'm setting everybody else up for harder questions to answer, which is kind of my job.
So I can't speak from experience. In fact, I have out of one, which Paul and Ben were involved with, where we used. But I think I worried about a lot as a clinician, right? Because it's theoretically possible, but we don't. I think this is where working closely with people who are discovering the phage and doing some basic work in the lab to see if it's possible. I think we've tried to be very careful and think about how to just avoid any risk to the patients, right? These are often sort of emergency use, FDA approvals, patients who are in pretty dire conditions. And so you don't have a lot of options, so you're already constrained. But if you thought that there was much of a risk of any harm, you probably wouldn't use it at all. And I think that's pretty much how I've let maybe Ben and Paul talk about this, but sort of avoiding any phage that shows these sort of warning signs. But I'd like to hear how whether or not you've had to make that decision on patients.
Yeah, definitely had to make that sort of decision. But that's like a whole separate thing, I think. But I guess specific for your question, how can we be confident? I don't know. We can have some confidence, right, from the lab stuff in how well we characterize the phage, right? So we have some phage where we sequenced, dozens to 100 of resistant mutants, and we see, oh, sorry, I'm echoing, and we see how many possible ways the bacteria can become resistant, like in mutations per gene, and we see, there's like 40 genes that can give it resistance versus, trying another phage in the library where we see, oh, there's like 4 genes and they're all related to the same, receptor. So maybe that one would be a better case, a better phage to use therapeutically. Another thing we can also benefit from is the diversity of phages, right? So if in the case you had, an antibiotic you use, and then all of a sudden now the evolved bacteria is resistant to 3 additional classes, that's a problem, right? Whereas if we have a phage that we use and it, becomes resistant to the phage, but we've killed off a bunch of the bacteria, we always have backups, right? So in the lab, we've got, here's our series of phages we can use, the next 6 courses we have planned out. Sometimes we only need the first course and then the person's fine. Sometimes you need to go to the second or third course, but there's always going to be phage that can infect the resistant bacteria. Like, I've never had a time when that's not been the case. It could happen, obviously, but we can always look back in the well, get new phage, characterize them, and use them. And so I think that helps with some of the confidence, but it doesn't guarantee that we're going to do it perfectly. It just gives us more options, I think.
Okay, that's great. Gosh, there's so many questions and hands up. It's getting a little bit confusing at this point. I just wanted to ask, do any of you who have your hands up want to respond to this specific question that Jesse asked?
Yeah, actually, I do, Charles. So quickly, I think what Ben just said is probably the thing, the diversity of phages, because in experiments we've done at A&T, and this is out of Dr. Liesl Jeffers Francis' lab, we found all sorts of correlations that result from anti-phage selection, and some of them make the bacteria a whole lot better than they were before the phage. So clearly, if you have a diverse phage library, you can try more and more phages and get one that isn't responding that way, because for any individual phage, you could get exactly what you don't want.
Okay, thanks, Joe. James and Randy, did you have a question related to that? If not, I want to ask Gustavo if he's still.
So let's see if Michael Hochberg is online, because his question, I think, is directly.
Yeah, I like that question too. Michael, would you like to bring up your point about competitive release, as you put it?
Yes, hello. Great talk and excellent discussion. So this relates to the first Joseph Graves question of replacing pathogens that are eliminated with more benign microbiota. And that is within the CF lung, there are lots of different pathogens that could be present. And the question is, if you're eliminating Pseudomonas aeruginosa, will other more nasty pathogens perhaps spread and colonize those spaces that are vacated by the therapy?
Yeah, great consideration, and I'm going to let the physicians answer this, or those who are closer to the treatment side. But I agree, theoretically, we know already proof of principle of that from laboratory experiments and a variety of biology. You sort of push something out, create niche space, and something else moves in. And if you have a system that is known to have this kind of, in this case, the CF lung, different points of the person's life, there's an expectation for what pathogens are going to be there or not. And a lot of that is driven by underlying competitive principles between those species as they come in and out. So on the one hand, very big concern that if you push a pathogen out, another one will enter. On the other hand, especially if you're dealing with emergency treatment, it's perhaps the last thing on our mind because the person is either undergoing organ failure, meaning the lung, or worse, threatened with death. So that's the kind of thing that becomes vitally important to track downstream. And that's what we're doing. But I hear you, it's a combination of both. You want to examine the problem, predict when it's going to occur, take longitudinal data, find out whether it actually occurs. And if it doesn't, that's more basic research to find out why. Did you perturb that ecosystem?
Yeah, the question is exactly ecosystem. So is there more of a community approach where a variety of phages like the ones that you've been investigating could be nebulized and eliminate parts of the community? And then relating to that first question, perhaps replace members of that community with more benign competitive microbiota.
Yeah, I think it would be exciting to study.
Yep.
Really interesting. Thanks for that, Michael. Gustavo, did you still have a question? I know you were very patient for a while, and maybe your question got asked by somebody else.
Hello, hi. First, thank you very much for a very good presentation. I want to ask you, I really like the idea of the trade-off, the evolution in the generation of the resistance in the bacteria. But I would like to ask a little bit more about the evolution in the phage. Have you seen changes in the phage during that process too?
Great question. I often get this question. Ben, I always keep asking you the hardest questions. I'm of the understanding it's kind of hard for us to find the administered phages, aside from maybe picking up the wild type phage that we kind of push into the system. Are you seeing much evidence from longitudinal sampling of phages that come out of the patients and that they are genetically different, or have we not looked hard enough to see if that's actually happened?
Haven't looked hard enough is the short. The long is it's harder to get them back out once you put them in, but we can definitely see the bacteria reduce quickly. But we have the samples banked sort of for sequencing. We could check that. Yeah.
Oh, Gustavo, we can't hear you. You're muted.
I was wondering if with time, it will be some process of selection too, kind of a arm race between the phage and the defenses of the bacteria, and you can have better phages in to kill or destroy that specific feature that the bacteria have.
Yes, so through in vitro studies that we're doing now with these phages that have been used in treatment or the focus of trials, our lab, James is looking at this in Georgia Tech. I think a lot of people are examining this question, and it's back to what we talked about earlier of an in vitro prediction. Will it match what you see in vivo? And we're excited to look and to really examine that, but it's an ongoing thing that we're pursuing.
So that's the topic of my CF fellowship and anatomy one I hold is exactly that question. What are the evolutionary co-evolutionary dynamics that are occurring inside these patients?
That's great. Okay, thanks for your question, Gustavo. And James, why don't you go ahead and ask your question? Or
yeah, I could ask Paul an email, I guess, but the question I've been thinking about a lot recently is that, this idea of the phage imparting selection. The CF example is that we have an opportunistic pathogen where these virulence factors, the type 4 pili, pyosin, they're not important for the infection in CF necessarily. How do you think this will translate when we have a true real world human pathogen, and we try and do something similar to this, where there's now selective pressure to, have some sort of phoenix mutation where this phenotype comes back?
Yeah, this is great stuff that I'm sorry we don't have time to talk about this topic fully. But what James is keying in on, I tend to think about it a lot, and folks in my group, is it's fundamentally different if you're trying to attack an opportunistic pathogen than one that is absolutely having to thrive in the host environment, right? A specific pathogen. So what I think helps our efforts, I will assert, is that when you attack opportunistic pathogens and you force them to give up or alter traits that are working in that environment, they have an escape route. They'll go live in the other environment for which they are already predisposed to utilize it. And that could be like soil or an aquatic environment for some of these pathogens. So to me, that's something that I apologize if it's been scrutinized to hell in the literature. I don't think it has. I think this is a great opportunity to consider what it is to develop this kind of therapy to target these kinds of pathogens, as opposed to if you were targeting something that is so specific that it has to be there. Is there a greater kind of opportunity to push things out of the niche when they have somewhere else to go? And again, I don't want to get into it too much because this immediately could lead us into a long discussion about SARS-CoV-2 or anything like in the virus pathogen realm, where we have a long history of, if that thing is only utilizing humans and it's something like smallpox disease and variola virus, it's a very different circumstance if it's something like influenza virus, which can exist in so many waterfowl around the world. You know, it's just kind of fundamentally different how you deal with these pathogen problems. So maybe I'm just bringing up a topic for a future EvMed, but I think it would be a fun thing to pursue more just conceptually.
Great. Okay. Last but not least, we'll be back to Randy Nesse. Go ahead, Randy.
Hi, everybody. I was going to ask a question about experimental evolution in phage and to what extent we could, I mean, there's a whole world out there of work that could be done if only there was enough money and people and time. But I look forward to hearing about that another time. We try to keep these Club EvMeds closing on just about the right time. I hope everybody here knows that Paul is now the incoming president for the International Society for Evolution Medicine and Public Health. And we all look forward to his guidance and leadership and creative suggestions for the society. So we'll look forward to seeing you in that capacity. But another Club EvMed on this topic, I think would be good in not too long.
Thank you so much for this
Thanks so
opportunity.
much, Paul.
And I appreciate that plug.
See you soon.
Take care, everybody.
Bye-bye.
And just to mention, next week, well, not next week, but our next Club EvMed will be Sid Tacker. He'll also be talking about antibiotic resistance. He'll be looking at it more from a One Health perspective. So, please join us for that one. But we do have to wrap up. Thank you, everyone, for joining us today. Thank you, Paul and Bob for participating, and Ben, who I know had to leave, and James as well. Great to see everyone, and we hope to see you at the next one. Great job.
Thank you.